Hello everyone, welcome to Bioverse. This is Suman, a biology lecturer who is here to make your work easier. See, the reason this video is made is to share you my experience in the evaluation center. As we get an opportunity to go to the evaluation center to correct the papers every year is a different experience. But this year, I would like to share certain important things which the new batch of students who is planning to write second PUC should be aware of. And also, all the students who have finished writing their second PUC exams, they are waiting for their results eagerly. They have told probably on 7th the results will be declared. But anyways, for the subject biology, the students have to know where exactly they have gone wrong. And the evaluation process is not that simple. It will be re-evaluated by the higher authority. If at all the students are getting more marks, further it will be re-evaluated. So it is not that easy to get 70 on 70, or it's not that easy to get above 65. So if you are getting 65 and above, then definitely yes, you have done your hard work. Every evaluator will be issued a scheme of evaluation. So the scheme of evaluation will contain the answers to the questions from the question paper, and also how much marks to be allocated to all the important points, and we will definitely look for a key terms to give the marks. If the key terms are not there, but you have filled the entire page of the answer that is related to that question, no marks will be allotted. Questions like multiple choice questions and fill in the blanks, almost all the students have done really well. See, I have evaluated I think around 189 papers. Out of 189 papers, I have found some 5 students who have got less than 21. That means failures are lesser only. Here and there, I found some mistakes that I thought, let me share it with all the students there so that they will understand where they went wrong, what to look for during the evaluation, and also students who are planning to write second PUC this year, it will be very helpful for you if you follow this. So 1 markers, we have no issues according to whatever the answers that has been given. The direct 1 mark was issued for them. Talking about 2 markers, see, I'm not going to discuss about every question here. The questions where most of the students have made the mistakes and where they could have corrected themselves, I'm going to talk only about that. See, part B, which is for 2 marks. Question is, draw a neat labeled diagram of mature 2-celled pollen grains. So 2-celled pollen grain here means a structure of a pollen grain that is having both the cells in it. As you know, vegetative cell and generative cell, these are the 2 main important cells. If you draw this diagram and label these 2 cells, then you get 1 mark each for the labeling. They are only asking 2 cells. If you guys have labeled exine, intine, germ pore, cytoplasm, vacuoles, you will not get any marks, because in the question paper very clearly they have mentioned label diagram of mature 2-celled pollen grains. So that means you are more specifically have to label vegetative cell 1 mark, generative cell 1 mark. That is very clear. Most of the students have done it, but I can say some of the students have made the mistakes with this. Such a simple diagram, but still mistakes were observed. Another important question where almost 75% of the students have committed the mistake is how ZIFT is different from IUI. Students have considered IUI, maybe confused IUI as IUT, and they have given the differences between the ZIFT and IUT. ZIFT is zygote intrafallopian transfer. So the zygote that is formed outside the body in the laboratory will be inserted into the fallopian tube. But here some of the students have not mentioned it is inserted into the fallopian tube, but with how many blastomeres? It will be less than 8 blastomeres. If it is more than 8 blastomeres, there is no need to introduce that into the fallopian tube. It can be introduced into the uterus. So students have not mentioned how many blastomeres for ZIFT. That is where they have lost half mark. If they have generalized and explained about ZIFT, they have got half mark. If they not mentioned about how many blastomeres, that means less than 8 blastomeres, they have not got the remaining half mark. And the another important thing is IUI. For IUI, which is intrauterine insemination, that means the semen is collected from the male outside and it is artificially introduced into the female body. This is insemination, which is done artificially into the uterus. And this is, where the students have got confused, considered this as IUT. So they are explaining about more than 8 blastomeres zygote will be introduced into the uterus. This is what most of the students have answered. That is where they've gone wrong. And another thing is, next question, pedigree symbols. That was the very simple, easiest one. Almost 95% of the students have got full marks for this. Sex unspecified, perfect. Whereas mating, male symbol, female symbol, in between these two symbols there will be a single line. That is mating. But some of the students have given double line. That is where they have lost 1 mark. Double line is going to depict that it is for consanguineous marriages. So consanguineous marriages is nothing but marriages between the family members. So that is where if you have done that, you would have lost the marks. Next question. The other questions are okay. Question number 26, that is, antibiotics acts as against life as well as pro-life to organisms. Here, some students have not attended this question only, considering it is very difficult. The thing is, this is so easy, but some have answered it. Intelligent students, they are very intelligent and very neatly they have mentioned, and I can say some almost 70% of the students, they couldn't answer this properly. Very simple one. Antibiotics are against life. It is against life means for bacteria and some microorganisms it is against. Okay. Pro-life means it is helping the living organisms to survive. That is for humans. Okay. So for human it acts as pro-life. For bacteria it acts as against the life, because antibiotics will kill the bacteria, whereas antibiotics, the same antibiotics is helping to sustain the life of a human. So that is the answer. 2 lines if you have mentioned about it. Bacteria, it is against life. Human, it is pro-life. If this is mentioned, at least you would have got some marks. But most of the students have got it wrong. And evil quartet. Next question. Very silly, very silly answers. Everyone knew about this answer, that is habitat loss and fragmentation, co-extinction, alien species invasion, over-exploitation. Everyone knew the answer. That I got to know when I saw paper. There is no student who has not attended this. Out of 189 papers that I have corrected, almost all the students have attended this, but only 20% of the students have got full marks for this. Where did they go wrong? Alien species invention, they have written, they have lost marks there. Co-extinction it is. Some of them have written coexistence, co-extensions. That is where they have lost marks. This is why the teachers will tell you, when once you finish your paper, read every line what you have written thoroughly, check thoroughly, and then submit the papers. So this is where students have lost the marks. Over-exploitation, over-explanation. Okay, this is what they have given. Overpopulation they have given. Okay, so some answers like this, they have lost marks. Please don't expect any minimal marks from this. Cut short marks will be given, and every marks will be allocated based on the scheme of evaluation what has been given. Okay, so this is with the part B. That's where the students have lost most of the marks. And part C, if you see, monocot embryo, any 3 parts, if you are labeling, 3 marks you will get. Very easily students have got marks. Placenta also, it's a good question. Yeah. And the next question, intrauterine devices, its mode of action. See here, this is for 3 marks. Any 3 mode of action you have to mention. You don't have to write any stories. Students have written stories. Okay. But I can say 50% of the students have done it really well. Remaining 50% they have written their own stories, and some of the students have written about the types of IUDs wherein they have lost marks. 0 marks were allocated. It is supposed to be phagocytosis of the sperm, decreases the motility of the sperm, make the uterine environment hostile. It is not going to be a suitable environment for the implantation in the uterus. These are the points. Just 1 or 2 lines it is. 3 points if you write, that is more than enough for 3 marks. So 50% of the students have written 2 points. One is motility, the other one is fertility capacity of the sperm is reduced. These two they have written. But regarding the environment of the uterus was not explained properly. Students have lost marks in that. And Hardy-Weinberg principle, students have written that well. Yeah, and disease causing organisms. Ascaris is mentioned. They are asking you the diseases, but some 30% or 40% of the students they have written for Ascaris, it is supposed to be Ascariasis. Okay, whereas students have written Ascaris lumbricoides. They are writing the full name of Ascaris. The species name also they are writing, and they are giving that as an answer. They have lost marks. Okay, and pneumonia spellings were wrong. Usually, see what happens in the evaluation center, the spellings are wrong. We are not going to cut marks unless it is sounding the same. Pronunciation is supposed to be same. Though it is a spelling mistake, but it is sounding like that word, we will give the marks. Pneumonia, if it is sounding, then we will give it. If it is not sounding like pneumonia, some other it is sounding, then we are not giving full marks. Bioactive molecules, yeah, most of the students have written this. Some students for Monoscus purpureus, instead of writing it as statin, maybe in a hurry, students have written stains, not statins. That's where students have lost marks. But these students who have written stains, no, they are not dumb students. They are good students only. Maybe they are in a hurry. They wanted to finish it faster. Maybe that's why they have lost marks in that. Then apart from that, 5 marks question coming to this mature embryo sac formation. Most of the students have lost 5 marks with this. See, they are not asking you to draw mature embryo sac diagram. It is not asked. What is asked? How that mature embryo sac is formed. This you need to talk about megasporogenesis. Okay, how the megaspore is produced. If you remember when your teachers are teaching, if you are smart and observing and understanding, 4 megaspores will be produced. Out of that, all the 3 will be degenerated, and 1 is going to develop further as embryo sac. How it is developed? The nucleus of that megaspore is going to mitotically divide and then produce 2 nuclei stage, then 4 nuclei stage, then 8 nuclei stage. That's how you are going to show how the embryo sac was formed. How 7 cells, 8 nucleus is formed. Which are those 7 cells and 8 nucleus? Towards the end you have to explain. There is no need for you to draw the diagram of embryo sac diagram. If you have explained this theoretically, 5 points, each point carries 1 mark. Most of the students have lost marks in this. If you are considering I have written this question perfectly, I have written 5 marks, I will get it, you will not get it. If you have written about the embryo sac explanation, then you will not get it. If you have written about the formation of the embryo sac, then you will get marks. Female reproductive system. Any 5 labelings you do, 5 marks will be allocated. Very simple. I think this is the first time it is, because every time students are supposed to, this question will be mandatory, either the female reproductive system or male reproductive system. Every year you can expect. So students had to write 10 labelings to get 5 marks, but this time they've made it much more easier by asking only 5 labelings. Almost all students have got, whoever attended this, almost all students have got good marks. Full 5 marks they have got. Then contact inhibition and metastasis. Students who don't know about it, they have written rubbish, but anyways other students have written the good marks. And talking about this treatment of cancer, see, this is for 3 marks. Treatment of cancer, if it is surgery, you write the heading surgery, but you need to explain how the surgery is done, what type of surgery it is, and just 2 lines about the surgery if you are writing, that carries half mark. And the heading surgery if you are writing, you will get half mark. But I think some 60 to 70% of the students, they have only generalized this and they have written surgery, immunotherapy, chemotherapy, radiotherapy. These are the treatment for cancer. If you have written, you are generalizing and if you are writing, you will get half of all these things, and you will get maybe for 3 marks, 3 headings will be considered as 1.5 mark. If you have given little extra details about every point, probably you would have got 3 on 3. Rest other as usual. Part D, as expected, the questions were asked little tricky, but if you know the concept very clearly, you would have definitely scored well. No issues with part D. If you know the concept, you have answered. If you don't know, as usual, you've written the wrong answers. But anyways, chromosome 16, sex chromosome gene disorder, the blood associated. Sickle cell anemia is what students have written, some of the students, but it is supposed to be hemophilia and thalassemia. Again, in thalassemia, more specifically, it is alpha thalassemia. But anyways, thalassemia, if you are writing about thalassemia, that carries 2 marks. If you are writing about hemophilia, you get 3 marks. Rest all other, I didn't find any issues. This is what I wanted to share it with you guys. Some of the questions wherein you have gone wrong, and students who are planning to write second PUC in the upcoming year, for you guys, make sure whenever any unit test is given or any normal test is conducted in your classes, or any board test, midterm, or any test or exam that is conducted, last 5 minutes you keep it for proofreading. You finish everything, last 5 minutes you do proofreading of your own writings. Every line, see, we are not robots to be perfect, we will definitely make mistakes. Every line from whatever the answers that you have written, read it. Okay, sometimes you might feel bored, sometimes you feel like I just want to give the paper and run away from this hall. This is all you feel, but you have to control your emotions and then read every line whatever you have written. Mark my words, minimum 2 mistakes at least you will find in your entire paper. If you are rereading your paper, you will definitely find the mistakes. Okay, so students who have done the silly mistakes, if they have gone through the paper once again, probably they would have written it somewhere at the end. Okay, and some students who have written repeated answers, see, some of the students will be thinking while evaluating, evaluators will not know, they will evaluate the same question 2-3 times if I am writing it 2-3 times. But honestly, I am telling you, every question we will check it. Even if you are writing 5 times in 5 different papers, we will write it as repeat. We will circle it and write it as repeat in every paper. And out of all the 4 or 5 times that you have written, we will only consider one where you have scored more marks, and that marks will be considered. If you feel that this exam you have not done well, you will also have exam 2 wherein you can try once again and you can give your best. Thank you all. Subscribe for more videos. I am planning to give you more and more information regarding the subject biology. Thank you all. Have a good day.